the gene pools of the Triticeae and the gene transfer from wild relatives. So you'll recall that we said when UG99 was discovered in the years that followed, we determined that the number of genes that were available to the community that were resistant to UG99, which is TTKSK, was a rather small number of genes, and so we had to start looking for genes wherever we could find them. And the easiest place to find them is in the primary gene pool. The primary gene pool is defined as those species with homologous genomes. These cross easily with common wheat, and so the D genome, you recall, is part of the A, B, and D genome of common wheat. That comes from Triticum aegilops tauschii. Triticum turgidum has the AADD genome, and there are, of course, a number of land races that are valuable genetic resources. And so those different progenitor species are considered to be in the primary gene pool. The secondary gene pool are those progenitor species with homoeologous genomes that are related to A, B, and or D. These cross with some difficulty with common wheat, but it certainly can be done. And so these are some of the genomes of species that are in the secondary gene pool. And then, of course, there's the tertiary gene pool. These species have homoeologous genomes that are not closely related to A, B, and D, diploid and polyploid aegilops species, Thinopyra, Cicale, and Hordeum. And they cross only with considerable difficulty with common wheat. Generally, if the cross is made, you would have to rescue the embryo, put it on a medium that supports an agar medium that provides all of the nutrients that enable the new zygote to develop into a plant that has some chance of producing at least some seed. Those people who work with these kind of crosses are very, very skilled, and I'm just not one of them. So regarding the wheat rusts, in particular, this publication, this book, really, Wheat Rust Analysis of Resistance Genes. I just wanted to make sure that you're aware that this is going to be in your packet of information that we're distributing. Bob McIntosh, Colin Wellings, and Robert Park were the authors, and as Robert said, that is now 20 years old, Robert, and we need to update it. There have been a number of genes that have been rust genes, both stem rust, yellow rust, and leaf rust, that have been identified and described, but that are not in this book. Chapter 1 is the wheat rust and the genetic basis of disease resistance. Chapter 2, the genes of resistance to leaf rust in wheat and triticale. Chapter 3, the genes of resistance to stem rust in wheat and triticale. And the genes for resistance to stripe rust. And if you're interested in some detail, if you look at page 6, right after the contents, there's a section on host pathogen genetics. It defines terms and provides some principles that will help you to study what you'll hear about next, which is what we'll be talking about, gene-for-gene interaction, and etc. Interestingly, in this book, there is a statement about the SR2 gene. And the authors offered the opinion that it is probably one of the most valuable genes that has been used in agriculture. So, discovering new sources of rust resistance in wild relatives. One part of the project was to look for resistance genes in other species. So these are the species that were evaluated. These were the number of accessions, for example, of rye that were evaluated for resistance to this pathogen, UG99. So out of those 533 accessions, 99% of them expressed resistance to TTKSK. For aegilops, out of 90 accessions explored, most of them showed some resistance to TTKSK. And so if you look here, the number of species that have some resistance is quite significant. Triticum timofeevi didn't seem to be very productive. If you looked at almost 300 accessions, you found only 15% of them that had a resistant reaction to that pathotype. So this is a little more recent presentation of the genes, origin and usefulness of cataloged and temporarily designated SR genes in conferring seedling and/or adult plant resistance to all PGT races belonging to the UG99 lineage. And so again, what you can't see here is a reference to a publication that Ravi Singh has authored. It has not been published yet, but we have it here for you to have this information. So these genes are ineffective. Here's the famous SR31 again. The rest of these genes are ineffective, but all of these are now effective genes for resistance to DTKST and other races that are in the UG99 lineage. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And we know this one is resistant to UG99, but if you recall our discussion about Digaloo and Robin, so although this gene is in fact resistant to the UG99 lineage, it's not going to do anyone in Ethiopia or Kenya any good. It may not do anyone any good in Egypt or in the pathway that we showed you earlier. From Triticum turgidum, here's the SR2 gene, SR13, SR14, and so on. So let me go back here and make sure you understand that these genes confer seedling and/or adult plant resistance. So does everyone have an understanding of the difference between a seedling test and an adult plant evaluation? So if it's a seedling test, these are done in the greenhouse with seedlings this big, and Sarala Sharma will show us some when we get to her lab. If you expose those seedlings to this lineage, and if there is no lesion or no infection, or one that is declared to have some resistance, then that gene is expressed not only in the seedling but through the entire adult plant. Whereas an adult plant resistance gene, we typically think that does not have an expression in the seedling stage. If you apply UG99 to a seedling that has no major genes, no seedling expressed genes, it will show that it is susceptible. But when you take that plant to the field and plant it and grow it and evaluate development of the pathogen over time, there is no development or very little development of the disease, and therefore provides protection in the adult stage of the adult life of the plant.